Dear students, new topic of lecture, cardiovascular system. It's first lecture from general part of histology, special histology. Special histology is a part of discipline which explain a structure of organs, system of organs. First of all, on its lecture, I want to say about types of organs. Organs to divide on two subtypes. It's first parenchymal organs, which include capsule, stroma, and parenchyma. Capsule is connective tissue composition, which cover of organ. Stroma formed by some parts of connective tissues, and in other words, it's a skeleton of organ. And parenchyma, it's a group of cells or just one type of cells which form its organ and represent functions of its organ. Second type has name tubular organs. Tubular organs always has cavity and tunics or special layers. Like tube composition in central part cavity, which can form some liquor, or if it's a respiratory system, it will be airway, and tunics, mucosa, submucosa, muscle, and adventitia or serosa. The cardiovascular system consists of the heart and blood vessels. On its slide present some main sentences which include in chapter cardiovascular system. Blood vessels take blood from heart to various tissues, and all vessels which provide from heart to tissues or organs are called arteries. Smaller arteries are called arterioles. Arterioles continue to capillaries, and then in capillaries to provide changers of gases. And next main sentence, blood from capillaries form collected and form venules, which are joined and form veins. Veins return blood to the heart. The cardiovascular system composed of two separate components. It's a cardiovascular system and lymphatic vascular system. Lymphatic vascular system formed by capillaries to start from ending parts, and no form circulatory system. Cardiovascular system is circulatory, and all blood will be circulate in organs and in general in organism. The first part of lecture, it's about composition of blood vessels. In general, blood vessels composed of three types of tunics: tunica intima, tunica media, and tunica adventitia. On its slide, you can see structure of wall, artery or vein. Look very attentive. It's represent composition of wall with tunics, tunica intima, which include endothelium. Next, tunica media, which include also smooth muscles, cells, and adventitia, which composed of loose connective tissue. On previous slides, I told about types of organs, and artery or vein, or in general blood vessels, are tubular organs because you see represent some hollow or cavity inside and wall composed of some layers: tunica intima, tunica media, and tunica adventitia. Tunica intima composed of simple squamous epithelium and subendothelial connective tissue. Endothelium composed of specific cells. Cells has name endothelial cells. Simple squamous epithelium, endothelium. In all blood vessels present simple squamous epithelium, endothelium. Tunica media, it's a middle layer of blood vessels and composed of layers of smooth muscle. Between smooth muscle layers, which represented by smooth muscle cells, present connective tissue sublayers with blood vessels and nerve endings. Smooth muscle cells lie circulatory, and between smooth muscle cells present more elastic fibers in artery and collagen fibers in vein. Tunica adventitia in blood vessels composed of connective tissue. Sometimes in tunica adventitia can visible some small blood vessels. It's a vasa vasorum, or another name, vessels of vessel. In large vessels, it's aorta or large artery or large vein. In tunica adventitia, can see in light microscope special small blood vessels. Arteries are efferent vessels that transport blood away from the heart to organs, to tissues, and arteries can be more large or more smaller, and in its case, in arteries may be present different types of hemodynamic and here on next part of lecture I will represent to you types of arteries. Classification of arteries. Arteries subdivide on three types: elastic arteries, muscular elastic arteries, and muscular arteries. And in different types of arteries will be represent different types of hemodynamic. On its slide, students, you can see differences between two types of arteries. It's elastic artery and a muscle artery or muscular artery. A visible difference in tunica media. And tunica intima, similarly by structure, it's endothelium and subendothelium connective tissue. And adventitia similar too, because present loose connective tissue, which includes some vasa vasorum elements. But tunica media is different, and differences joined with names of its arteries and different differences by hemodynamic. In tunica media of elastic artery present elastic fibers more, appositive of smooth muscle cells. In muscular artery present more smooth muscle cells and not more elastic fibers. Elastic arteries include aorta or large arteries which lie near of heart, and in its arteries present very high blood pressure and very important elastic wall of its vessels. And in its case, in aorta or large arteries present more elastic fibers. And in a period of high blood pressure, wall not destroyed and can change position. Next, muscular artery composed of more in tunica media smooth muscle cells which lie circulatory. It's a very important to increase blood pressure more far from heart. Position of muscular arteries always more far from heart, and main function of muscular artery is increase blood pressure by contraction of smooth muscle cells. On its slide represent information about differences between elastic and muscular artery, and I want to say some words about muscular elastic artery or elastic muscular artery. It's a middle to concern size of arteries, lie more far from heart, but after elastic part of arteries represent middle by size artery muscular elastic, which include in tunica media together smooth muscle cells and elastic fibers. And name of its artery joined with amount of elements. If it's elastic muscular artery, more elastic fibers. Muscular elastic can be present more smooth muscle cells and not more elastic fibers. In muscular artery also present specific elements of elastic fibers, I told on previous slides, and very important specific structure is elastic membrane. Elastic membranes represent in muscular artery its internal elastic membrane and outer elastic membrane. Internal elastic membrane lie between tunica intima and tunica media, and outer elastic membrane which lie between tunica media and tunica adventitia. Students, it's a good slide, show structure of tunica media in aorta. You can see more elastic membranes, wave shape, and maybe present from 50 to 60 elastic membranes in tunica media. It's a specific element and structure unit of tunica media in aorta. Muscular artery include smooth muscle cells in tunica media. Here you can see on its photo from histological specimen, and also present small blood vessels in tunica adventitia. You can see vasa vasorum. And here are visible internal elastic lamina and outer elastic lamina, which lie between tunica media and tunica adventitia. Shape of internal and external elastic lamina similar to elastic lamina of aorta or elastic membranes of aorta, shape with waves, and its waves can form after death of organism. It's a very important because in process formation of histological specimens, it can be after death, and on slates, histological slates, in arteries, or always you can see in tunica media, between tunica media, tunica intima, some element of waves. It's the internal elastic lamina, and also internal elastic lamina will be provide waves in tunica intima. It's a photo from histological specimen. You can see small artery and small vein. Well, very nice visible differences between two vessels. In small artery present more thick layer of tunica media with smooth muscle cells, and in vein more thin layer of smooth muscle cells. It's a great difference between two blood vessels, and I will say about its differences more in next part of lecture on next slides. Arterioles. Arterioles lie more far from heart and muscular artery continua and form arterioles. Arterioles near in diameter maybe 50 micrometer, and in arterioles present smooth muscle cells and not visible internal and external elastic lamina. In arterioles which lie near of a muscular artery, present maybe some 2 or 3 layers of smooth muscle cells. But in arterioles which stay near of capillary, can present just one layer of smooth muscle cells, and distance between smooth muscle cells more far. And in final part of arteriole disappear smooth muscle cells, and arteriole continue to capillary. On its photo of histological specimen, you can see differences between arterioles and venules to concern composition of wall. In arterioles present smooth muscle cells in wall and wall more thick, and diameter of arteriole more smaller appositive of venule. In venule, diameter more large, but wall very, very thin and not present smooth muscle cells in wall. Arterioles continue and form next part of cardiovascular system. It's capillary plexus. Capillary plexus include different types of capillaries. In some parts of organism can present somatic capillar or fenestrated visceral capillar or sinusoids. And capillaries continue next part to venules. Sometimes present specific element is anastomosis, arteriovenous anastomosis. It's a good scheme present in your textbook. Visible arteriole and next capillary plexus and postcapillary venule. Here are information about different subtypes of anastomosis. Can form anastomosis between arteriole and venule, and its anastomosis may be represented by just one capilla, it's number 2 in its picture, or meta-arterioles part. Also present specific capillary plexus between two veins. It's maybe represent in liver, and present glomerulus capilla. Glomerulus capilla can lie between two arterioles, and it's maybe represent in kidney. On previous slides, I told about different subtypes of capillaries, and capillaries subdivide to concern structure and location in human organism to some types. The first I want to present to you continuous capillaries. Continuous capillaries more smaller and diameter approximately 8 micrometer. Wall of capillar formed by endothelial cells which lie on basal lamina, and in outer part of basal lamina present specific perivascular cells, pericytes, and present reticular fibers. Capillaries, continuous capillaries present in next part of organism. It's skin, it's bones, central and peripheral nerve system, and in its case, its capillar has name somatic capilla because present in soma element. Fenestrated capillars, second type of capillars, and in endothelium present specific apertures which cover of diaphragm. It's a very thin cytoplasm of endothelial cell, and its diaphragm and apertures help to provide transports of elements. And fenestrated capillaries present in kidney, in endocrine glands, and in more specific types of glands which provide and help transports of elements. On its slide present scheme composition of capillar. It's fenestrated capillar because visible fenestra, its apertures in endothelial cell. Black points on its picture. Basal lamina cover and form a wall of capilla. It's electron micrograph section of fenestrated capilla. Why fenestrated? Because you see screws show fenestra in cytoplasm of endothelial cell. Covering of endothelium and also present visible basal lamina, nucleus and organelles, cytoplasm of endothelial cell. On slate information about location, continuous capillaries stay in skin, connective tissue, muscle, lungs, brain, bones, and I told continuous capillaries in some book has a name somatic capillaries. Fenestrated capillaries present via in renal glomeruli in kidney, in the intestinal villi, endocrine glands, pancreas, and fenestrated capillaries also has name visceral capillaries. Next type of capillaries has name sinusoids. It's a very large capilla by diameter, near 20 micrometer, and a very, very thin wall of capillaries. Present fenestra in endothelium and specific structures pores in basal lamina. It's a very open capillaries. Sinusoid capillaries present in liver, in red bone marrow, and in organs of hematopoiesis, spleen, and it's a very open capillaries, I told. About arteriovenous anastomosis. It's a special part of blood supply because arteriovenous anastomosis, it's a structures formed without capillary, arterioles joined with venules, and capillaries not present in its part. It's a very important structure because it can increase blood pressure in venules part. It's a first. Second, will be provide more quickly blood from arterioles to venules, and anastomosis helps to thermoregulation in skin, or maybe helps in provide some functions in spleen also. Arteriovenous anastomosis formed specific combinations, I told, and maybe form special structure is called a glomus. Glomus cover of connective tissue. Each glomus consists of afferent artery, one or more coiled S-shaped connecting vessels, and efferent vein. Wall of afferent artery includes more smooth muscle cells, which form more thick layer. In other words, it will be sphincter of arteriovenous anastomosis. After contraction of smooth muscle cells, can close of arterioles and not provide to continue blood to anastomosis. Venules continuous and joint and form more large vessels vein. Vein, similar to artery, has three main tunics: tunica intima, tunica media, and tunica adventitia. On next slides, you can recognize differences between veins and artery. And please look very attentive on its slides and recognize main differences. It's a very important. You can use slides and form conspect about differences between artery and vein. You can divide your paper on some two parts and write differences artery and vein. It's help to you to practical class. On its slide, very nice visible differences between artery and vein, muscular artery and muscular vein. You can see size of tunica media. It's a great difference. Size of tunica media in artery more larger appositive of vein. And very nice visible difference in tunica intima because tunica intima and tunica media, I told in artery, between its two tunics present special internal elastic membrane. In vein, its internal elastic membrane not present. To concern size of adventitia, you can see in vein adventitia more largest, more thick, appositive of artery. Next slide is about classification of veins. Look, on its slate presents the first type, it's venules or small veins, diameter near from 15 to 20 micrometer. It's a very small blood vessel, wall very thin, and surround wall of very thin part connective tissue. The next type is medium veins, diameter near 1 centimeter. And tunica intima includes endothelium, reticular fibers, basal lamina. Endothelium always lie on basal lamina or rest on basal lamina. Next part, smooth muscle cells may be present, and in its case, medium veins to subdivide on two types: muscular medium veins and non-muscular medium veins. Muscular medium veins include smooth muscle cells and collagen fibers, fibroblasts, and non-muscular medium veins not include smooth muscle cells. Or only collagen fibers and or fibroblasts, it's the cells of connective tissue. And third part, tunica adventitia, more thick and includes few blood vessels. Will be vasa vasorum. The next type is large veins. Large veins return blood to heart. It's a very large vessel with large diameter, and in wall of large veins present three tunics, very nice visible, and more thick part of tunica media. But please, students, remember tunica media in large veins, smaller appositive of tunica media in artery. The heart, very important part of cardiovascular system because heart is a very important organ which provides bloodstream and provide blood pressure to blood vessels. Heart is composed of three main layers. It's endocardium, myocardium, and epicardium. In heart present cavities, four chambers, two atria, and two ventricles. Students, information about structure of cardiovascular cells or cardiomyocytes you can see in previous lectures about muscle tissue. Please look very attentive and all elements of cardiomyocytes and process contraction of cardiomyocytes. On its lecture, I want to say about specific element of heart. It's special elements or special cells which form conducting system of heart. What does it mean, conductive system? Conducting system provide autonomical contraction of heart. It's in other words, it's automatia of heart. Heart conducting system composed of specific cells. It's cardiomyocytes too, but its cardiomyocytes not contract. Look, the first type has name pacemaker cells. Cells controlled of contraction of heart. In adult, it's near 70-75 beats per minute in not active position, not active state. And pacemaker cell more present in sinoatrial node and provide contraction 70 beats in 1 minute because in itself present regulation of ions and form in result depolarization of plasma membrane. Second type of muscle cell, in modified cardiac muscle cell in conductive system, it's cells which form atrioventricular node more, but in atrioventricular node also present pacemaker cell, and a modified cardiac muscle cell, second type, formed atrioventricular bundle or bundle of His. It's a cell similar to pacemaker cell, but not form very active depolarization of membrane, 70 beats in 1 minute. It's maybe 30-40 beats in 1 minute without pacemaker cell. And atrioventricular cells, which form atrioventricular bundle, very important in regulation contraction of chambers of heart. Third type of conductive system of heart has name Purkinje fiber cells or specific Purkinje cells in heart. What does it mean, Purkinje fibers cells? Purkinje fiber cells, specific cells, very large by size, very nice visible on light microscope because cells more large, nucleus stay in peripheral part and cytoplasm formed a light pink color and present more glycogen inclusions in its cell. In cell not present more myofilaments and cell if it's light staining area similar to adipocytes, but be attentive in adipocytes in central part not present something and because remove of fat and here in Purkinje fiber cells nucleus stay on peripheral part, light staining of cytoplasm and present more glycogen inclusion. Purkinje fibers composed of 2 or 3 layers of cells. Position of cells Purkinje fibers in histological spacement between endocardium and myocardium. Students, it's a very good scheme present in your textbook. You can recognize yellow color on its picture, main elements of conducting system of heart. Atrioventricular node, next, bundle of His, sinoatrial node includes pacemaker cell, atrioventricular node includes second type of muscle cells of conducting system, and some cells from pacemaker cells. Bundle of His composed of second type of cells and Purkinje cells, so Purkinje fibers. You can see third type of cells in conducting system. Students, thank you for your attention. Here is a good picture. You can use recognized main elements of cardiovascular system.